current diet. Are we adapted? Do you think we are adapted to the current diet? Look at the image I have put here. It seems to be an apple, but maybe what we are not eating is precisely an apple. Nowadays, there is a very high prevalence. I would say there is a pandemic, even more than the pandemic that unfortunately affects us today, of chronic diseases such as obesity, diabetes, hypertension, dyslipidemia, and other chronic diseases associated with nutrition. The nutrition we have today. And this entails such profound environmental changes that lead to a change in human behavior, which evidently results in a change, the diet, in the diet we have today. But we have to think that humans have a genetic structure and that in these environmental changes we are introducing, it may be that we do not adapt to this nutrition and diseases arise, which we are now going to talk about. The crux of the matter, or the basis or the root of the problem, is what is called evolutionary discordance. What is that? Well, precisely, we have a genetic structure that does not balance with the diet we have today, and it leads to the diet we have today, and not only from 10,000 years ago until now, we have a genetic structure that does not align with the current diet and leads to the development of chronic diseases, as I mentioned before, and others. And not only 10,000 years ago, but since the Industrial Revolution, about 100, 150, or 200 years ago, which has made a significant turn in our way of eating that does not align with our genetics. And what is this evolutionary discordance? I will give a brief summary so that you understand how our way of eating has changed from the first humans to the present. The first humans were called Homo habilis, and they were gatherers. What does this mean? They ate what they could find from the earth. That is, their diet was predominantly plant-based. 87 to 99% of their diet consisted of plant products. Then they became scavengers, meaning they ingested dead animals, and they started discovering that they could eat the bone marrow from the skeletons of dead animals. What did this mean? The overall energy intake in their diet increased significantly. This energy intake increased their brain mass, and in this way also shortened their digestive system, typical of carnivores. Carnivores have a shorter digestive system, but a slightly larger brain system. Then came, well, this was the Paleolithic diet that is often talked about lately. Is it an ideal diet? The Paleolithic diet consisted of 37% protein, 40% or 41% complex carbohydrates. Now I will explain what complex carbohydrates are. 22% fat. This was the ideal. What happened? Then came the Cro-Magnon man. He was smarter, already had weapons to hunt, and became hunters. These hunters were expected to have a diet composed of at least 50% animal protein, such as. But when agriculture and livestock began, approximately, as I mentioned, about 10,000 years ago, and there was our problem because in agriculture and livestock, we could now plant, for example, wheat. We already had mechanisms to grind the wheat and sift the flour. And that meant that when we sifted the flour, at first we ate everything that was from the wheat, including the husk and the germ, which we must not forget. That is where the micronutrients are. But as it evolved, we could sift the flour and left what is called the endosperm. The endosperm is starch. There are no micronutrients. This caused human height to be shorter because we were not providing everything we needed. That was when the evolutionary discordance and the electrogenetic alteration began. That is when what is called genetic adaptations occurred, such as insulin resistance and leptin resistance. But we still had an advantage. What was the advantage? That we still cultivated the land, hunted animals, which meant we exercised, allowing us to compensate for the excess calories from cereals. The most pressing problem came with the Industrial Revolution. Look at the image below. Only 5 generations have produced disease in humans because our genetic system has not adapted to our current diet for 10,000 years. What is the characteristic of the current diet? Well, as you know, it is a hypercaloric diet. It has a high calorie density, and perhaps in a very small piece of food, we have a lot of calories. It also has a high proportion of saturated fats and trans fats. And what are trans fats? Trans fats are fats that are not natural and therefore can lead us to develop many diseases, many metabolic disorders. But the proportion of omega-6 fatty acids has also increased at the expense of omega-3 fatty acids. And I will explain later the importance of these polyunsaturated fatty acids, the omega-6 fatty acids, which in our current diet are flooded with omega-6, are pro-inflammatory. What does this mean? They are pro-disease. And there is also a decrease in the intake of complex carbohydrates and fiber, both insoluble and soluble fiber, which insoluble fiber will ferment in our intestine, producing short-chain fatty acids like acetate and hydroxyl butyrate. And these are essential foods, energy for our microbiota. Don't forget this. Our digestive system is very important, and our microbiota is even more important. So this will nourish our microbiota and will nourish our digestive system. But there is another problem, as you can see here. Modern man tends to gain weight due to this hypercaloric diet, due to the increase in fat, which will lead to a mismatch in the controlling mechanisms of hunger signals. And where do hunger signals come from? We will see this later. They come from our adipose tissue. Thus, all of this reaches Homo sapiens sapiens, who have very little of sapiens because we are harming ourselves. And besides the change in macronutrients, we have a change in micronutrients. Look at the image on the left to see how it has evolved from hunter-gatherers to today. In micronutrients, for example, how vitamin C has decreased. Vitamin C, if you don't know, I will tell you, we need to obtain it from food. We are not capable. There are animals that can synthesize them themselves. We are not capable of synthesizing them. And vitamin C is involved in many biochemical processes, especially in the control of oxidative stress. The same goes for vitamin E, which you can see has dramatically decreased. Total fats increase, saturated fats increase, omega-6 increase, omega-3s decrease, and starting from the industrialization stage, non-natural fats, trans fats increase. But as you can see in the image on the right, there are 4 that control the entire nutrition industry. And what do? They make hypercaloric foods that are highly palatable. What does highly palatable mean? That they are pleasant to the taste. And how do they do that? By adding a large amount of fat. And this contributes a lot of calories and leads to disease and inflammation. And as I put down here, we must conclude that a calorie is not simply a calorie. There are many colleagues, friends who tell me no, because to control not gaining weight, I roughly count the calories and see, and so on. Yes, I think that's very good, but not all calories are equally important. I will give you an example, and I think you will understand it very well. Imagine that 500 calories are 4 tablespoons of olive oil, which along with a healthy diet, many papers tell us, decreases the incidence of heart attacks and strokes. However, a sugary soda from today is only 150 calories, but it is associated with the development of type 2 diabetes. Therefore, a calorie is not simply a calorie. We must take into account what produces that calorie. Indeed, right? But we must take one thing into account, what is called biochemical individuality. What does this mean? Each human being has their own biochemical footprint, a unique profile, tension, a unique profile of nutrient requirements that must be met to maintain health, otherwise chronic degenerative diseases will arise. What does this mean? That each of us has our own genetics and what is called genetic polymorphism, which can even be a change in a single nucleotide of our gene. That is what is called SNPs, and it is what will give us our biochemical individuality. Imagine, I'll give you an example. There is a change in an allele in a gene called FTO, which is a gene associated with obesity. This obesity-associated gene has 1.67 times more likelihood of causing obesity in those heterozygous patients who have this polymorphism. So imagine how important biochemical individuality is, as it can be perfectly different from the neighbor I have next to me. That individuality plus the effect of the environment, that is epigenetics, which includes diet and exercise, can lead to the development of disease. What is epigenetics, or what is the effect of the environment? That is to say, each person has a diet that should be individualized. This is what is called nutrigenomics. That is to say, nutrigenomics is how foods affect the development and expression of certain genes. So each of us, depending on certain foods, will have the possibility of developing or not developing a disease. And what example can I give you? Imagine that you eat a diet high in simple carbohydrates. This will lead to insulin spikes. And as you will see later, this causes inflammation. Moreover, if you also eat foods with a high amount of omega-6 fatty acids, this will also produce inflammation because it will produce cytokines called eicosanoids. These type 2 and type 4 eicosanoids are inflammatory. If I also have in my genetic code the probability of developing an inflammatory disease, the environment, which is the diet or lack of physical exercise, will cause that gene, which is dormant, to express itself and potentially develop an inflammatory disease. Therefore, the diet should be individualized through nutrition and, of course, exercise to prevent the development of disease. This highlights the very important biochemical individuality of each person. And there is a dietary balance, as I have mentioned, in fats. Notice that the ideal consumption of omega-6 to omega-3 should be 4:1, but nowadays I have noted the current consumption is 25:1, and I would say it is even 35 to 40:1. There is an excess of omega-6. And let's not forget, I repeat, omega-6s are inflammatory. They can produce, as I mentioned here, inflammatory diseases. And we will see this a little later. What does all this translate to? I am telling you, silent systemic inflammation or low-grade inflammation. What is that? When we get a bruise, we experience inflammation, an inflammation that is clinically visible with certain clinical characteristics. For example, there is heat, redness, pain, and functional impairment. That will not be the case. In this silent systemic inflammation, we will not notice anything. And that depends on this unbalanced concentration of PUFAs, which are polyunsaturated fatty acids, are in cell membranes and act as fuel for the genesis of oxidative stress. And let you know, if there are many health professionals listening to me and those who are not, the genesis of oxidative stress and oxidative stress is the basis of any disease and, of course, of aging. As you can see here in the image, it is an iceberg. The whole problem is that we see nothing outside, but the problem is inside. And just because we do not see what is inside does not mean it is not harming us. Here I briefly present the chain of omega-6 and omega-3. The head of omega-6 is linoleic acid, and of omega-3 is alpha-linolenic acid. Through a series of enzymatic processes of elongation and saturation, what are the different substances is produced. Omega-6 ultimately converts into arachidonic acid, which we can also introduced through animal fats, not only through omega-6, but from the fats we have today. That is the animals we have in the past. Early humans could have livestock that they already controlled, but they grazed. Right now we have them caged, we have them eating cereals, mainly corn, which has drastically changed the fat proportions. In the past, there was a high amount of vitamin E, a high amount of polyunsaturated fatty acids, and now they are saturated fatty acids, mainly arachidonic acid. And as you can see here, they are inflammatory eicosanoids, which make us sick. However, the omega-3s we have here, the most visible one is eicosapentaenoic acid, which is EPA. And another one is docosahexaenoic acid, which is DHA. These are anti-inflammatory because they will ultimately produce eicosanoids called protectins and resolvins, meaning they are the firefighters that will extinguish the fire of this inflammation caused by omega-6. Therefore, our cell membranes should be saturated with omega-3s rather than omega-6s. But today it is not so. And I will explain how this problem arises. The mechanism of action of inflammation. Here we have an image where we see the outer part, that is a cell membrane, which is a lipid bilayer composed of phospholipids, polyunsaturated fatty acids. It also has some cholesterol, but fundamentally polyunsaturated fatty acids. We will expand on all of this in a much broader way so that you understand everything in the course that, God willing, will start on the 29th. But well, broadly speaking, if we have that membrane saturated with omega-6 fatty acid, any stimulus on these membrane receptors called toll-like receptors, usually 2 and 4, and these stimuli are often simply trauma, infections, stress, which no one today considers the capacity that stress has to make us ill, sudden temperature changes, and LPs. What are LPS? They are called lipopolysaccharides, which are the membrane structures of gram-negative bacteria we have in our microbiota. I insist again on the microbiota, the importance of the microbiota, and this will cause that toll-like receptor to be expressed, and that phospholipase 2 begins to perform the different functions of desaturation and elongation, thus forming arachidonic acid. Here, lipoxygenase acts, forming leukotrienes, which will cause inflammation, especially at the pulmonary level, asthma, and similar conditions, and cyclooxygenase, which will produce prostaglandins, prostacyclin, and thromboxans, that is, inflammation and high coagulability. And well, I will tell you more about it later. This is inflammation. Now I will explain a little about what LPS is. The LPS, as I have said, are compounds from primarily gram-negative bacteria, which enter our intestine through food, mainly through a diet high in animal fats and proteins, but animal fats. What does this mean? Here you see the lamina propria in our intestine. These LPS are capable of stimulating the toll-like receptor of the lamina propria and stimulating our innate immune system. That is to say, it calls macrophages to produce pro-inflammatory cytokines in defense of that bacterial product. It is not something that our immune system recognizes. Additionally, as you can see in the image next to it, in our lamina propria, you will see our macrophages and our dendritic cells from the Peyer's patches that are on the lookout for precisely everything bad that is there, and they will detect the LPS and also stimulate this immune system. But LPS can also be bound to kilomicrons and enter the vessels of the intestine, producing what is called metabolic endotoxemia. And this metabolic endotoxemia is capable of producing chronic inflammation. Moreover, not only that, this metabolic endotoxemia can directly reach the liver and produce what is called non-alcoholic fatty liver. And non-alcoholic fatty liver does not always have to be associated with obesity. I have patients who are, in quotes, thin and have fatty livers, and it is related to that alteration of the microbiota with that dysbiosis, and that chronic inflammation due to the stimulation of an intranuclear transcription factor called NF-kappa beta will produce insulin resistance, dysbiosis of the microbiota, insulin resistance due to inflammation, and as you can see in the image on the right, how important the affectation of the mucosa and all these structures is. This inflammation will be able to cross the blood-brain barrier and will be able to produce neurological disease. Look, there are autopsies of patients with Alzheimer's disease that have been found to have, besides the amyloid substance typical of Alzheimer's, these LP compounds that are capable of crossing that membrane, that blood-brain barrier, and producing cognitive decline and disease. And it will produce a decrease, which I present here as a small detail that I will expand on later, a decrease in BDNF, which is a brain-derived neurotrophic factor and is associated with proper functioning of the brain. So notice the inflammation, the connotations, low-grade inflammation, the very important connotations it has.